hacer el mismo link. Espérame. ¿Te mandé el link de la transmisión o no a YouTube? Creo que se lo mandé al Jorgito. Ya, lo voy a probar, te aviso cualquier cosa. Chau.
All right. ¿Está esto? ¿Aló? Hola. Hola, 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 hola. Aló, aló. ¿Ahora sí? Ok. Gracias. So we are back. Gracias por volver. So we're starting a little bit late. I was not, and I'm pretty bad with distances. And the Institute for Biological and Medical Engineering is sitting in the middle of the campus, and this is at the entrance. And I never thought of that distance, walking distance. But we're back a little bit late, but we will catch up afterwards. Okay, so we already understood a little bit about plastics in terms of accumulation, what they can do with human health and also with the environment. And now we are going to move after a great presentation by Beatriz, which was the last one from the morning. We'll move into the more biotechnology side of things, which is what we can do with the enzymes that we're finding that can also degrade some plastics. And we will be delighted by a talk by Wolfgang Zimmermann. And I have to mention a few things about him. So we communicated something about 5 years ago. And probably both my research in this field and the research that Bea and I are doing with the metagenomics wouldn't be possible if it wasn't for a very nice intention from Wolfgang to collaborate after we met. I visited the University of Leipzig, in which Wolfgang is located before the pandemic, and we were discussing a little bit about how to work with plastic degrading enzymes. And he's an expert in the field, and he's been working with this for a very long time. Most of the enzymes that we know have been characterized by his group, and with some remarkable, amazing results. So he became professor of and chair of microbiology and bioprocess technology of Leipzig University, especially because of those reasons. Very outstanding work on degrading plastics. And he's now professor emeritus at the Institute of Analytical Chemistry in the same institution since 2019, so right before the pandemic. So we've been talking about you coming to Chile for a while. I'm delighted that you're actually coming, that you're already here. Well, yeah, just the stage is yours, and thank you for coming.
Yeah, good afternoon, and thank you, Cesar, for the nice introduction. As he said, we had a very nice and fruitful cooperation in the last years. One of his students came to finish her PhD. We have another student from his lab. He's in Leipzig now. And we did a couple of nice papers as well. So, as he said already, we're going to focus a little bit on applications, on solutions, possible solutions. Not for all types of plastic, but for one type, which is actually an important one, specifically in terms of recyclability. Now, the talk is about synthetic plastics, but I will focus very quickly on polyester type plastics. But we already heard this morning, there are different types, and very important one is the CC carbon type, polyethylene, polypropylene, then there are also polystyrene, PVC, and so on. The difference is, as has been mentioned also before, the structure of the backbone, which on one type we have the CC backbone with very little options to modify this backbone chemically or even less biologically. And then we have the group of the heteroatomic backbones, and here we find the polyesters like PT or polyurethane. When it comes to enzymes involved, what we know so far, we have to say we know not too much about enzymes which could modify or degrade those polymers with a CC backbone. We have some research going on showing the involvement of redox enzymes like lactases or peroxidases, phenol oxidase has been reported, among others. When it comes to applications, we have to be realistic to say we are pretty far away from any industrial applications of one of these enzymes. The story looks different when we come to polyurethanes, at least to one type, which is called a polyester-based polyurethane, simply because we have some ester linkages within the polymer, and we are able to attack that with enzymes. Most of these enzymes are the same, which work on polyester like PET. We have an urethane linkage in the polymer, which may require the presence of another enzyme, an urethanase. And recently there has been some progress in identifying specific enzymes for degrading this linkage. We also did experiments years ago with polyurethane foam, for example, what you find on your hiking boots and so on. It's a big problem what to do with all these plastic boots. They are bought, the shelf life is very short. The kids buy it for fashion reasons, and after 3 months they end up somewhere, and it's a huge pile you cannot imagine, and you cannot separate that properly, you cannot recycle it. So that's an issue. If we can develop the technology further to address polyurethanes, at least the polyester type, which is a big part of the PO business. And then we come to the ones I'm going to talk more about now today, and this is the polyesters, most prominently PET. And here we have a couple of enzymes I'm going to talk more about it, specifically the cutinases, which are our favorite enzymes we're going to work with, and I show you why is that. So the good news is that polyester type polymers we can actually attack, we can modify and degrade using enzymes. And among these, the most prominent one is, of course, polyethylene terephthalate or PET, but also other type of polyesters like polyethylene furanoate, which has been promoted as a substitute for PET. Polycaprolactone is another one. Polylactic acid can also be degraded by these enzymes. This has an application in, for example, agricultural films, now made most of them with PLA. So the degradation of that is also of interest. And then we have mixed polymers like, for example, polybutylene adipate copolymers with terephthalate, which is again more easy to degrade by enzymes or in the natural environment. And as I said before, PU based on ester linkages. So I'm going to focus on PET. And when we have a look where it comes from fossil resources, of course, which is part of the problem, either from natural gas or from crude oil. It goes to a reformer and cracker stage, and on one side you end up via ethylene glycol. This is one of the monomers, and on the other side, through the reformate and BTX, you end up with terephthalate, and this is then synthesized in a condensation reaction, a polycondensation reaction composed of these two monomers. Some points to remember: it's a thermoplastic polymer compared to others which have other properties when you heat and cool them. We should also remember that this polymer contains both amorphous and crystalline, or let's say semi-crystalline regions, which is quite important when it comes to enzymatic degradation of it. So why is it actually resistant to degradation? And the two reasons are, first, the crystallinity, and second, the presence of aromatic constituents in the polymer chain. In this case, it's the terephthalic acid, of course. So this makes it difficult for enzymes and for microorganisms to degrade that sort of material. The products we all know. On one side, packaging, food applications. We all know our plastic bottles, they're also present here. And the other large application is in textiles. And I'm going to show you applications for both of these areas where we could make use of enzymes to modify this material or maybe even degrade it in the aim of recycling it. Why do you want to do that with enzymes? Why do we want a biocatalytic approach? The reason is we have done research which shows that it is possible to use these enzymes for an environmentally friendly functionalization of textiles, especially PET fibers. But functionalization means we just want to modify properties of these fibers or this polymer. We don't want to degrade it. And the second one is now, after some years of research, we are able to have very powerful enzymes which can not only modify the polyester but actually break it down to the monomer in a reasonable amount of time. And then, of course, we see how can we apply that. We can apply that to fight the plastic problem and also to make a contribution to recycling, which is then non-chemical. Yeah, we've seen this slide before. It shows the interest in the field, not only in the research community, but also in the public, in the media. We see the number of publications is going up very steeply in the last years. Also, we are able to discover new microbes which are able to produce these enzymes, or maybe which are able to degrade the material itself when they're exposed to it. And we also get our hands on more interesting enzymes, which are both robust and powerful enough to be applied in an industrial environment. The number of enzymes able to do that is growing. I can say every week you may find a new publication about a new strain which has been identified. And I'm gonna talk a little bit about what is common about these enzymes and what is different. First of all, we could say they are all esterases. That's the truth. More specifically, they are all ester hydrolases, and the majority of them are serine hydrolases. So they have the same catalytic triad, the same reaction principle. Is very similar to all of them. So you may say, if you go in nature, you may be able to find esterases which are able to hydrolyze not only their natural substrate, what they have been designed for by nature, but they're also able to deal with polyesters. But this is mainly due to a low substrate specificity, or let's say substrate promiscuity. And this has to do with the architecture of the active site. I will show you that in a minute. A subgroup of these ester hydrolases are cutinases. And I'm going to talk more about those because the enzymes we work with mostly come from this group. So these are natural occurring enzymes, and they have been designed by nature to degrade plant polyesters. So in plants, we find polyesters which are looking a little bit similar to our synthetic polyesters, and we take use of that and apply these enzymes, again, using substrate promiscuity to hydrolyze chemically man-made polyesters, such as PT. And then there is a second group, which is a little bit distant. These are carboxyl esterases. Basically, it's also esterases, same reaction mechanism, but they are specific on hydrolyzing PET oligomers or small sections of the PET polymer. And the so-called MHET-ACE is also belongs in this group. So this is an intermediate product, the MHET, we already heard about it, together with another one, which is called BHET. This is the favorite substrate of these carboxyl esterases. Now, talking more about cutinases, As I said, it's a plant polymer. It's an aliphatic polyester. You find it in all aerial parts of higher plants covering the surface. It has the function to protect against loss of water, diffusion of water, and also it's a protection barrier against pathogens. Another related substance is superine. Superine is also a polymer composed of aliphatic and aromatic monomers. Again, hydroxyfatty acids. It contains ferulic acid and other phenyl propanes. And again, that makes it look similar to the substrate we're dealing with. PET, you remember we have the aromatic moiety in the molecule. And here you see the similarities in the structure. This is the structure of suberin here, and you have to imagine this as a three-dimensional structure within the cell surface of the cell. This is the primary cell wall, and here you have this lamella, and you can recognize some structural features which make it look similar to PET. Also in cutin, you have also some aliphatic esters. Well, looking closer at the cutinases, we find them all over in very different habitats. Both fungi and bacteria are able to make these enzymes. All of them are esterases, alpha beta family, hydrolase members. They all have more or less the same catalytic triad composed of serine, histidine, and aspartate. They don't have a lid. If you're in the enzyme research, you may know that lipases, the other group of esterases, have a lid, but these don't have a lid, and this is of advantage when you want to hydrolyze a long, complex polymer, so there's no hindrance to enter the active site. They have a broad range of substrates, which has to do with their low substrate specificity. They can hydrolyze low and high molecular weight esters. Again, this is what we need. Actually, you can also use them for the reverse reaction. You can use them to synthesize polymers. This is another area of application. I'm not going to talk about that more, but this is something you should also keep in mind. You can use these enzymes for synthesis reactions. This may be interesting when we want to construct novel types of enzymes like we heard this morning, engineering polymers in a way that they are more easily degradable. And then such a controlled esterification reaction may be an interesting option. The other interesting thing is that most of them are thermotolerant or maybe even thermostable. And when it comes to the complete degradation of PET, you need to have thermostable enzymes. So you can do something with mesophilic enzymes or with a cold-loving enzyme, as we heard in the morning, but not for the degradation of, complete degradation of PET on an industrial level. There may be other applications, and I'll tell you that in a moment. So what do they do? As I said, they play a role by the during the attack, the initial attack of phytopathogenic organisms like fungi and bacteria. When they attack the plant cell, they have to go through the cutin or maybe even the superine barrier. How they do this? They secrete cutinases, and in this way they get access. Here you see the hyphae here attaching to the surface of the plant, and the hyphae goes inside because it can dissolve the cutin or even the superine layer. If you have a potato, the potato peel is also containing superine. That's a brown color. If you leave that on your kitchen too long, you see a fungus growing. And how it gets in? He has a cutinase to get through the potato peel. In this case, we may call it a superinase, but it's the same idea. A second very important function is that these enzymes play a key role in the mineralization of plant matter, in the recycle of carbon in the environment. Otherwise, we would have ended up drowning in leaves and tree plant products if they are not degraded, and cutinases play a very important role here. Now, the notion that it's actually possible to use these enzymes to modify or degrade synthetic polyesters goes back a couple of years, and it has been the group of Wolf-Dieter Deckwer in Braunschweig who worked on Thermomonospora fusca, which is an actinomycete, a thermophilic actinomycete. We can find this everywhere. Actually, we found our actinomycetes just in the compost outside of our campus, so we didn't have to go to fancy places to find these things. So they noticed that aliphatic aromatic copolyesters can be degraded by this enzyme. Now, this type of polyester is a little bit more easy to degrade because of the aliphatic sections within the polymer. So this gets more easily degraded than PET from a bottle, let's say. And then in 2004, we put out a paper where we actually showed that the same type of enzymes from the same type of organisms is able to modify the fibers made, textile fibers made from PET. So that was our research focus at that time. We haven't been thinking about recycling. And our favorite microorganisms, which we studied for many years, is also Thermobifida, which we isolated. One of my students isolated that from a compost in her garden. It's a moderate thermophilic. It lives in soils and composts. This one produces two isoenzymes, turned out to be two cutinases, very similar properties. And actually it produces a second enzyme, and this is one of these carboxylesterases. So these two work together when it comes to modifying or degrading polyesters. An important difference to the PETase reported from Idunella is that these strains here, or let's say all the cutinase producing strains, they cannot use the degradation products. They don't have the biochemical pathway to utilize ethylene glycol or terephthalic acid, which also points to the fact that their ability of these enzymes to degrade PET is not a natural function. It's like an additional function we have. Luckily, we see that. Serendipitously, we found it is possible to use this function to degrade synthetic enzymes. But it's not an enzyme which has been evolved due to the presence of plastic in the environment since 1950. This enzyme has been around for millions of years. Right, looking closer at this production strain, this Thermobifida, we found that it's a very interesting and versatile producer of polyester degrading enzymes. And we studied a couple of them, not only the cutinases, we published that already in 2006, but also the carboxylesterases are interesting. They also produce hemicellulose degrading enzymes such as silonases and mannanases. Here we resolve the structure of a mannanase from this microorganism. They also produce cellulases, and they also produce redox enzymes like peroxidases, which may be interesting. Nobody has looked at that yet, but this type of microorganisms are pretty interesting for biotechnical applications, I think. Now, in 2014, we solved the structure of the cutinase from the Thermobifida, and this allowed us to have a rational design approach on creating mutants, or generally better understanding the function of this enzyme when it comes to a synthetic polyester like PET. This is one slide showing the reaction products of the carboxylesterase. So that's the second group I said. I think it's they play together in the degradation of this material in nature. But when you isolate this enzyme, you can do other interesting things with it. For example, you can degrade these cyclic trimers. These are compounds which are an unwanted side product in the polycondensation reaction. When you make the PET, they tend to deposit on the surface of the fibers, and they create a problem when you make a textile out of it. It means you cannot color the textile, the fibers in the way you want. You get some of grayish color, a big problem in the textile industry. What they do is they use organic solvents to dissolve this out before they do the dyeing, but this is not environmentally friendly method. And so that's actually one of the applications of these enzymes to use in the treatment of textile fibers to remove these trimers. But they're also doing other things. If you feed them a trimer or a dimer, they will break that down to the BHAT, MHAT, this is the monomer, and then eventually to terephthalic acid. And you can use that as a sort of synergistic enzyme together with a cutinase to speed up the degradation of a PET. Yeah, this shows effect of a treatment, of a surface treatment of a fiber. Here you have, this is a SEM, you see the deposit of these trimers on the surface. And here you have like a cleaning step with a rather low activity of the enzyme, but it's enough to wash off the unwanted trimers, and you get a clean surface, which is then used for textile dyeing. There are other applications actually where you have not a fiber, but you have a film, and you want to deposit something on the film, for example, aluminum, which is used for sunshades and stuff like that. And again, with this enzyme method, you don't need a chemical treatment of the surface. You have an environmentally friendly way to do that, and there are companies interested in that. Right, let's look at similarities or differences of some of the polyester hydrolases which have been reported so far. And you notice immediately, even if you are not expert in molecular biology, that they all look pretty similar. This is the Thermobifida fusca enzyme. This is another cutinase-like enzyme from the metagenome, which is now applied in an industrial process. And this is the PETase from Ideonella. Now, the differences are basically in the architecture of the active site. And you can see that in case of the PETase, this active site is a little bit broader. And this is an explanation why this enzyme works at a lower temperature. It's more flexible. It can open up more easily, accommodating the polymer chain, which goes in here somewhere, at least to some degree. I mean, to understand why you need a higher temperature, you have to know about polymer chemistry a little bit. So the architecture of the polymer strain, of the polymer backbone is that it has both an amorphous and a crystalline part, as I said. And the amorphous part is sort of individual strings of polymer sticking out. And the fact is that at a low temperature, let's say ambient, or let's say in the Arctic water, this will be rather stiff material, and it will be very difficult for the enzyme to reach that, to reach the individual, let's say, strings of polymers. So some hydrolysis may occur. Certainly, nothing will happen with the crystalline part. So you can get a partial hydrolysis at lower temperature, but you will never get a complete degradation at lower temperature. Right, this paper has been mentioned already. We published that a couple of years ago together with a group in Hamburg who did this hidden Markov modeling, which showed us very nicely that right now we are only looking at a very small fraction of what is actually around in nature. And at that time, we found these two groups, the thermophilic ones containing the thermobifida. Here are the fungal ones. Fusarium solanae is one of the cutinases which has been studied many years ago in parallel to the, no, not in parallel, a couple of years before the actinomycete cutinases were described. Again, this was for other applications, but it's the same enzyme. They already mentioned here that there may be a huge group of other polyester hydrolases, some of them from mesophilic sources, or also from cold environments, including marine sources, and that really has been the starting point to look at things like Moraxella here, or Oliospira, both of these strains which have been worked on in Chile here. Recently, just 2 weeks ago or so, I noticed this paper. I'm not sure if you have seen it. It just shows that these enzymes now popping up from everywhere. And this one has been isolated from human saliva metagenome by a group in Thailand, actually. They report in Angewandte Chemie. And if you compare that, the MG8, that's the one from the saliva, if you compare that with the petase or even with the Fusca cutinase, it's not that bad. It has quite some activity here. Again, I'm wondering, isn't that just a polyester ester hydrolase, which by some luck has activity on the polyester, synthetic polyester as well. Anyway, I think we will see a lot more coming up. There has been another paper recently from McKean's group in the UK and from the American guys. So the question is, yeah, maybe you've seen the movie. Where could we find more of them? Do we need more of them? The answer is yes. We have always two problems. The thing is, do we have the high activity enough to put that in an environmental context if we want to have an enzymatic recycling process? And is that thing stable enough? Industrial application mean it's rough environment. There may be stirring or mixing or pumping. Does the enzyme survive that? Another problem is, and we don't have a solution for that, maybe there is no solution. What about the crystalline part? So far, there is no report convincingly showing that the crystalline part of PET can be degraded. I haven't seen any. When you want to degrade crystalline PET, such as the PET bottles, you have to have some sort of chemical or physical pretreatment. Otherwise, it doesn't work. So where to find them? We have two approaches. One of them we heard from Gabriele this morning is the old-fashioned cultivation. We use environmental samples. We cultivate a strain, isolate a strain if we are lucky, then make a functional screening, and finally got the enzyme. This is how we started, and we were sort of we were in a good position that we worked with actinomycetes because a lot of research in the 50s, 60s has been gone into the discovery of new antibiotics. And from that, we knew a lot of, let's say, secret culture conditions to pull out the more interesting ones. Otherwise, you always get the same stuff, or you may not get it at all. But you can get some interesting stuff, and we put that in our culture collection. But this is not enough. We also used a metagenomic approach. And here we started with something like a compost. We isolated the DNA, the metagenomic DNA. We put in a PCR in the degenerate primer method, where we matched that with known sequences containing some variability in some of the positions of the bases. And that allowed us to identify groups of hits, a vector library, which then got through an activity screening, purifying the enzymes. We actually then tested all of them on the real substrate, which is a PET, not on some model substrates. And finally, characterize them. And by doing that, we got hold of a number of very interesting enzymes recently. And again, this is just the tip of the iceberg, as I call it, just looking at cuteinases. There's a huge biodiversity of these. This is a number of them just reported last year by Liang and co-workers. The green ones here you see, these have already been confirmed PET degraders. Some of them degrade PCL or PLA. So there's more to come from this side. And this shows the result of a very small and modest sampling foray into compost heaps around our university. And we found a couple of enzymes which we reported recently. We call them PHL. So these PHL ones are very likely to be from the same group of actinomycetes, close to Thermobifida. There are some other relatives here. And you see this quite away from Ideonella. When we looked at the properties, we were interested in two things, activity and thermostability. Then a couple of them sticked out, like this one, the number 7. And you see it's quite a huge difference in terms of activity and also in terms of thermostability. This compares to the strain we've worked for many years before. And this one is, of course, much stronger. Just to repeat the hydrolysis products, when you incubate this enzyme with a PET substrate, you end up with basically two compounds such as terephthalic acid, this got stuck somehow, terephthalic acid and ethylene glycol. Intermediate products are MHET and BHET, but if the reaction is taken long enough, or if you put enough enzyme, you go straight to TPA and EG. So you don't need MHET aces or something. You just need one enzyme to go from PET to the monomers, which makes it easier for a technical application. Yeah, if you compare that to the known, or let's say to the one which has been the number 1 before, we found that, the LCC, you can see it's much stronger and it produces TPA and via MHET in a very short reaction time. The weight loss, so we measured the weight loss of a plastic disc, is also remarkably faster with the PHL7 compared to the red one here, the LCC. So this has been published involving Paula from Chile, also Felipe is there, and of course Cesar. We made this paper together, it's a nice one. Then recently we solved the crystal structure of this enzyme, and I'm not going into details, just showing, even though it's very similar to the ones we already know, the performance is much better, the activity is better, the thermostability is better. Why? There are very small changes, differences in the active site and also in sites which you don't expect, which are pretty far away from the active site. We call it subsites, and the architecture of the subsites have a strong influence on the ability to efficiently hydrolyze the polymer chain. For example, this L210 is one area where which plays a role in this high activity. So that means when we look at new ones, we don't look at a completely different type of enzyme, but we look at, let's say, a variation of the theme, and there can be surprises. There will be surprises. We cannot predict 100% with rational design what would be the ideal enzyme. So it's really going out, having luck, and if we're lucky, we can find an interesting one. Now, this is how we think it works. Here you have a film, a thin film. If you throw that the enzyme in, the enzyme attaches. It has hydrophobic sections on the surface, which made it stick to the surface. Here, and then we have a subsequent degradation, in this case of both sides, of course. How the enzyme works, it cleaves a couple of monomer sections of the chains of the amorphous chain, and then subsequently these trimers or dimers are split down to the monomers, ethylene glycol and terephthalic acid. And you can follow that also on SEM. Here you can see after a couple of hours you have these little holes and craters showing up. And we looked at it with vertical scanning interferometry, and here you see very nicely how the enzyme is digging little craters inside the material. Why is that? We think this is reflecting areas of different amorphous and crystalline regions which are differently attacked by the enzyme. And even more though, if you do this experiment also with the same technique, here we cover the surface so the enzyme cannot work, and then we look at the surface retreat, how it's eating into the surface of the film. And again, you see the LCC, compared to the LCC, the PHL7 is much faster and much more rigorous degrading the film. Now, we still need to do more. This is natural enzymes we find in nature, but we still need to do some genetic engineering to push them further to where we need them for technical applications. And as I already said, we have to focus on increasing stability to be able to react at a higher temperature, because higher temperature means maybe even above the class transition temperature, which is around 70, may be favorable for a complete hydrolysis. And then the second is we want, of course, high activity. The higher, the better. And this may be not the best one, which is around. Maybe there's another one, which is found next month or in 2 months. We have a lot of tools available. I told you we started with rational design. Once we got the structure, we could do that. There has been a directed evolution approach. There has been a paper published by American group. Recently, there are a lot of data-driven models. Machine learning is becoming very popular to design ideal PET hydrolases. So there's a lot of things going on at the moment. Now I want to talk a little bit about potential applications. And this one, the first one is in the textile industry, and this is how we started. As I said, at that time, we used enzymes which were not strong enough to break down PET completely, but to, let's say, modify it a little bit. And the enzyme is introducing functional groups on the surface of the polyester, thereby changing properties like hydrophilicity, which is, again, a problem in textile. If you have a synthetic textile, you notice that the transport of water is not good. You start sweating. If you have a wound dressing, you don't have, it doesn't take up moisture. To get that, you need a chemical treatment. Again, this is a rough treatment. It degrades the quality of the material, and it's toxic for the environment. So we have been looking at environmentally friendly methods to increase the hydrophilicity and to improve the textile properties. There are other properties you can improve. For example, the dyeability of such a treated material is different and right now has to be achieved by rough chemical methods. And here you can see if you put a drop of water on the untreated material, it doesn't go in, just stays there. After enzyme treatment, the material becomes hydrophilic and absorbs the water straightforward. Another interesting application is in the laundry industry. We have been working with a laundry company to develop enzymes they put in the washing powder to wash textiles. There are problems with synthetic textiles in the washing machine. They tend to form little pills. The color is modified after some time. As a result, you have to throw away your t-shirt earlier. It doesn't look nice, the colors anymore. If you treat it with the enzyme, it has a longer lifetime. The brightness of the color is staying for a longer time. Also, you can use less detergent, and you can wash your textile at a lower temperature, which is also interesting for energy saving and for the CO2 footprint. Here you can see the control textile forming these little pills after some time on the surface. And here with the enzyme treated, the material stays clear in the soft surface. So we filed a patent for that a while ago, and this enzyme is presently developed. And this is actually one of the applications I see for these mesophilic enzyme, or even for the psychrophilic enzymes, because here you need an enzyme which is not ripping through the polyester like the butynases, but is like softly modifying the surface of textile fibers, for example. Could also be textile films. There are interesting applications for printing on the surface of PET films. For example, for the electric industry, you can clean the surface using this method. And the enzyme, which will be described by Cesar in a couple of minutes, is one of them. I think that has the potential for that. Now, this one you've seen in the morning already. It just shows that the plastic has becoming a problem since 50. We've heard that. And related, there has been a problem in our environment, especially in the oceans, but also in landfills. And it is becomingly considered a threat also for human health. We don't know what happens with the plastic we inhale or we take into our body. And this is a paper from me showing that there are a lot of ways microplastic or nanoplastic can invade our body. It can go through the blood barrier. It can actually reach any organ of our body and create inflammation and do other things, which we don't know yet, but we don't really want that, do we? Now, this is also a very popular sketch here showing the core of the problem. The amount of plastic produced, we had heard this morning, and the amount of problem discarded. Some of them is incinerated, at least it's not going in the environment, but the amount of recycled one is a rather thin line. And this is what we have to do. We have to increase the amount of recycling, recycled PET. We have to increase the use. We have to go away from one-time uses of plastic. And we have to think about other feedstocks. Is it possible to have plant-based feedstock, go away from the fossil feedstock? Using enzymes in this recycling process can help us in a way. It will not be the only way. There's chemical recycling, there's mechanical recycling, but I think enzymatic recycling can find a place there. On this basis, we suggested in this paper in Chemsos Chem, which has become pretty popular in the last month, how we can completely degrade a specific type of post-consumer PET plastic at a reasonable temperature, about 70 degrees, degrade it to the monomers, and then at the same time use the monomers to resynthesize fresh virgin PET. And you can do that basically forever. There is no decrease in the quality because you go down to the monomers and then you go up to the PET again. And we could show that we can do the whole circle. Right, of course there are problems, and some of the impediments I've listed here. One of them is continuing low cost of fossil-based PET, even though we have a oil and gas problem at the moment due to the war in Russia. Even of other reasons, fossil oil and gas is still cheaper and has to compare with the price of recycled PET you have to acquire on the market. And there's actually a shortage of recycled PET. That means the price goes up. And then using this as a starting material makes it economically not feasible to do that. This can change, of course. It has to do with the availability of sampling infrastructure to collect a lot of different types of plastic, which can be processed in one way or the other, chemically or biologically. Then we are competing with the non-enzymatic recycling methods. This is always a threat, but we have some advantages when it comes to specific streams. I'll show you that in a moment. A limitation may be that for some of these streams we are looking at, we right now don't have the collection infrastructure. We do have in Europe at least a very good infrastructure for collecting PET. We all have to pay a deposit when we buy a PET bottle. This is almost completely 100% collected and recycled. For other streams, this is not the case. So if that changes, a lot of other things would change as well. I already talked about the limited availability of the feedstock, but again, if the collection system is improved, this may change as well. And another problem is the partial crystallinity of the feedstock. Now, hydrolyzing a PET bottle with these enzymes is still not possible completely without any pretreatment, and this is of course an impediment. Yeah, this is to illustrate that. We have inside the polymer, we have a lamella, and these lamella are composed of both amorphous layers and crystalline layers. There may be some amorphous parts sticking out which are easily digested. Other parts cannot be reached, especially not at a lower temperature. And there's really no way at the moment around it other than having a pretreatment. And here you can see this is a paper we did a while ago in Advanced Science. It shows a direct correlation between the degradability of the material and the initial crystallinity. The higher the crystallinity, the less you can degrade with the enzyme. So very clearly that is a problem. This is a slide about the competition. Of course, we have mechanical recycling, which is very active and which is not sleeping. It's developing at the moment very fast. We have now very advanced recycling plants which can produce food-grade material. And this is, of course, potentially cheaper than an enzymatic process. It has disadvantages because, after all, you lose the quality of the material. It's a downcycling, as it's called. The polymer chains break up. You have to mix it up with virgin PET to maintain the function. And then after another go, it may end up in the deposit or being burned. Chemical recycling is another threat. There are a couple of ways to chemically recycle PET. Some of the problems here is you can only do that at a very large scale to work profitable. Otherwise, it is just not competitive with the mechanical recycling or even with biological recycling. But again, a lot of research is done at the moment, and there are new chemical catalysts developed which allow a more efficient conversion of this material. So this is quite a serious competition to our biological process. So you can see there's a lot of options here: hydrolysis, alcoholysis, glycolysis, chemical ways to do that. A lot of big companies are investing in that or opening plants for that. And here we have biocatalysis as one option of a chemical catalysis. Basically, it's the same. We just don't use a chemical catalyst, we use a biological catalyst. Yeah, but what speaks in favor of the enzymatic process? It's a green process through and through, so we can sell that. The companies love to sell that. on their label saying we recycle PET with a green process. We don't need chemistry. We use that at the ambient temperatures. We go back to the monomers. We can do that forever if we have the process. And the other advantage is we can also handle post-consumer PET, which cannot be handled by the chemical or mechanical recycling. And this is the so-called difficult to recycle type, for example, multi-layers, which means packaging, for example, which is composed of different layers, composed of different plastics. You cannot throw that in a chemical recycling plant. It will kill the catalyst. You cannot recycle that mechanically. That means it goes in the landfill or the ocean, or it's burned, so it's gone. Mixed plastic waste, this is the reality from household collections. They're always mixed. So how to do with that? Do we have to separate that, or can we have a selective degradation of the PET part? The enzyme is quite tolerant to contaminants. There may be contaminants brought in from the collection process, or there may be additives within the plastic which is killing the catalyst. Another one which I think is important, it's a scalable process. A chemical plant or a mechanical recycling plant, you cannot scale down considerably. So you have to have an infrastructure to collect the bales. You have to transport it by truck or ship or whatever. And then you have a central place where you have the big plant. The biocatalytic plant you can have at a small capacity anywhere, basically. You could think of it as on a small island in a developing country or in a remote place where they still drink water, but they don't have the plant to process it chemically or mechanically. So instead of throwing it in the ocean, you can have a little plant to take care of that. And then we identified one waste stream which is actually possible to completely hydrolyze at this time without any pretreatment. And the pretreatment is a major bottleneck in making the whole process profitable or economically feasible. This is just a slide to show you how difficult these multi-layer packagings are. So a lot of engineering has been gone into construction of these layers. So there are up to 10 layers, for example, for food packaging to maintain the freshness, to avoid oxygen coming in. This is nicely engineered, but it's not very good for deconstruction and recycling. Basically, this is burned. Whatever you collect for recycling, this is not recycled, this is just burned in the best case. In the worst case, it ends up in a landfill. We have been working on these multi-layers also in a EU project recently, where we actually took those multi-layer packagings and selectively were able to remove the PET, and we ended up, usually it's a bilayer, so we have a PET layer together with a polyethylene or polypropylene. Treating with the enzyme, we can get rid of the PET, and then the PE is recycled alternatively by chemical recycling, pyrolysis, for example. So we've done that with some major PET producers like Indorama, and with some packaging producers like Kreiner in Germany, or Suprema, a French company in polyurethane synthesis. Yeah, what are, you can show these challenges here. How could we make this process more profitable? This is a scheme of the process which is presently established on a pilot scale by a French company. And basically it shows here a pre-treatment step, which is a very energy consuming. You have to actually melt the PET bottles. You have to put in a lot of energy. After that, you extrude it. And by doing that, they even have a grinding step here. By doing that, you convert the crystalline part into the amorphous part. And then the amorphous PET is hydrolyzed by the enzyme. That's one bottleneck. And what we have been proposing is When we get rid of the pretreatment, we save a lot of energy, and that makes the process much more favorable. What they do right now, the terephthalic acid they produce has about twice the price as the terephthalic acid from fossil oil, so nobody would buy it. So how can we make it cheaper? One way is get rid of the pretreatment. Second one is try to get a better enzyme, make it faster, let it operate at a higher temperature, and this is the second area where I would focus to make this process more profitable. Looking at alternative streams of post-consumer plastic, we know the PET bottles, very well established in many countries. Plastic packaging has a major contribution. We heard that also in the morning. A major part of plastic waste comes from plastic packaging, especially in the food industry. And we identified this type of packaging, which is called thermoforms, which you find everywhere in the supermarket to wrap your vegetables, grapes, for example, or eggs. This is material made usually from recycled PET and or these clamshells, as they're called. The nice thing is they have a low crystallinity, about 6 to 7%, and our enzymes can fully eat them. There's no pretreatment at all. They have quite a large market share. It's not a fringe market. Presently, they are just wasted. They are burned or landfilled. We can do that now at a low temperature reaction. We don't have to add any chemicals. It works at the aquasystem, and the whole thing has a very small carbon footprint compared to let's say, chemical methods I just talked about. So this is what we use. This you can actually chop it down to some pieces. You can also throw in the whole bottle. I will show you a clip at a moment, and you will end up with basically terephthalic acid and ethylene glycol. We also did that on a reactor scale, and here you can see the terephthalic acid concentration goes up, and the Eg concentration goes up, and then there has been an intermediate MHET concentration, which is then going down because all the MHET is also converted to terephthalic acid and Eg in the end. So you only end up with these two compounds. You can filter off the terephthalic acid because the solubility is very low, and you can distill off the Eg. Then you have both components. The market value at the moment is at the terephthalic acid. That's the most valuable thing. But basically, you can use both monomers and reuse them. So this is what we suggested here. Using thermoforms, hit them with the enzyme, get back to the monomers, and resynthesize it to normal PET. And here you see a little clip. What we did, this is a reactor, about 1 or 2 liter reactor, where we have been feeding the whole thermoform package from the supermarket at I think like 60 degree or 70 degree, and you can see the little watch here. What happens after 16, 18, 20 hours, 24 hours, it's gone. And then we throw in another bottle, another clamshell, and it's gone. And you can do that for a while. So you don't need a lot of enzyme, and you end up with the pure monomers. Right. This shows some of the people who have been involved in this work, both from my former lab at the microbiology and bioprocess technology, and then since the last 2 or 3 years at the analytical chemistry lab. Paula Sanchez has been joining us. There's also people at the institute like Chen Song and Patricia who work with NMR technology to study the function of these enzymes. And I thank you for your attention.
There's time for a few questions if there's any questions from the audience. But I'll start by making one, which is, does it ever get inhibited, the enzyme, after feeding it with so much plastic in those experiments?
We have done, we did experiments with another enzyme, which is not that powerful as the PHL7. And we noticed when we do that in a closed reactor, you actually do observe some sort of inhibition coming from the intermediate product MHET, because they are competing with the active site. So the enzyme has to do, the enzyme has to do, it's working now. The enzyme has to do two jobs. Now it has to hydrolyze the polymer, and then one time it has to hydrolyze the MHET down to terephthalic acid, and it cannot do the same at the same time. And we solved that by taking out the MHET from the reaction by a membrane. But this enzyme we have now doesn't care. There's no inhibition. There's not, let's say the MHET is not accumulating in amounts that would make a problem. Even though it's a closed container, it's a batch reaction, but it's not a problem. There's no inhibition.
Okay. And then my second question, I'm going to take the opportunity that nobody's raising his or her hand. My second question will be whether, if you were, I'll translate you outside of Germany, and I'll put you in a different context. So in a challenging scenario in which you can do research, but you don't have that much funding, okay? If you had to choose between doing sampling compost, plant compost, and then searching for the enzymes, or doing what is now taking place, which is a lot of protein engineering to try to, make these enzymes even better, what will be your tool of choice? Would you prefer to still search for novel enzymes in other plant compost elsewhere, or would you decide to jump onto the computational and experimental designing part?
I think ideally this should be, or this needed to be combined. What I wouldn't do is what has been done with the PETase, that you take an enzyme which is completely unable to work at a higher temperature and try to kick some stability into it by making all sorts of modifications, extra loops and blah, blah, modules. In the end, you end up with an enzyme which is very similar to an enzyme you can find in a compost heap next to you. So especially if you don't have a lot of money, I wouldn't suggest that. It has been done. People have been investing amazing amounts of money to do that, basically to show that, yes, you can convert a PETase into a functional active polyester hydrolase, but for what price? So I would say going out to nature and tapping into biodiversity is still a valuable option, because we can just not predict what we can find. It seems very similar, but there are things which we don't understand yet, specifically the importance of the binding, the importance of the hydrophobicity of certain sites, which are quite away from the active site, and which we don't know why, but it works. It has a big difference. And the best thing is to let nature find out what they have developed, because these enzymes are very suitable for a broad range of substrates. But then you get something which you still want to improve, and then the genetic engineering kicks in, and then maybe you are able to improve the thermal stability by a couple of degrees to make it even better. Yeah, but I wouldn't. You have to combine these two.
Hello. Hello. Well, again, my name is Gabriel. I wanted to ask, the reaction you showed in your video, that was of an entire like intact packaging, food packaging. When it's shredded, does the reaction speed increase, or it's just or it's about the same?
Well, basically it's a surface reaction. So if you increase the surface, you would get a quicker reaction. So if you like, you can mill it and probably this will speed up the reaction, but you don't have to. The I think the company, they use cryo milling or something to grind it down. You can even do something more sophisticated, but it's not suitable for an industrial application. You can dissolve the whole PET and then precipitate it, and then you get nanoparticles with a huge surface or a material which is very porous, and this one gets degraded even faster. Okay. But it means additional step, it means additional cost, and here I have been looking at the economic feasibility because we want to compete with terephthalic acid or with PET made from fossil fuel. So it has to be cheap. That's why it should be easy, okay. And we don't need to have any grinding or milling or whatever.
Okay, and well, my second question is. You mentioned that, well, these multi-layered packagings and with different types of plastic that are difficult to metabolize. Have you ever run into some problems with proprietary compounds? For example, you in that infographic, in that picture, you showed some trademarks of DuPont, for example.
Yeah.
Have you ever had any problems with them not being like forthcoming with their chemical formulas or something like that?
No, the problem is that they are very secretive about the composition of their material. We would be very happy if we know what they throw in there. Yeah. And recently there has been a paper that showed that I think 200 compounds are inside it, and we have no idea what it is and what it's doing to, for example, the environment or to the human body. This is a very big problem. These companies hired very able chemists and engineers to produce the most durable and functional material, but they have not been thinking at all about what happens to it after it's used. And this is a big problem. So really, I think we have to open it up, make it transparent, and make them change the way they produce this material. It has to be more simple. We have to go away from this sophisticated multi-layer. Then they will say, hey, but when we do that, then what happens? Our product in the supermarket will spoil very quickly, and we have to throw away the whole food. Isn't that worse for the footprint? Isn't that worse? You know, I think it's a wrong argument. I mean, we have been living for thousands of years without that material. We bought the fresh produce in the market. Nobody suffered from that. I mean, we had losses that stuff gets spoiled, but we don't need these high-tech layers. They are very nice and functional, but they are not designed for recyclability. That's the problem. That has to change.
All right, well, we thank again Wolfgang for a nice presentation. And we're gonna take a quick 15-minute break. I think there's some food outside, I'm not sure. We can check, but we're gonna take a quick break and then we come back for the last two talks. thanks to Wolfgang, there are enzymes that are known to degrade this synthetic polymer, which is a PET. And there are other accessory enzymes that can degrade the byproducts of the reaction. But overall, we have seen a bunch of actually enzymes from different environments and from different organisms and some uncultured bacteria that can degrade this plastic. And just to briefly mention, for those that work on biochemistry or that like proteins as much as I do, it was nominated as the molecule of the month from the protein data bank where all of the protein structures are stored in January this year. So Wolfgang already mentioned that there are many different enzymes. They have been discovered in a very wide span of years. So since 2005, there were the first mentions of different enzymes that can degrade plastics. And here in the Y scale, you have like just different rates of degradation. And this came up during the conversation, so that there might be with different enzymes with different efficiencies. And we might want to know how efficient they are before working with them in industrial setups. So if you can see from here, you also have like a blue to red gradient, which means what's the temperature that these enzymes use. So most of them are thermophilic, and this was already mentioned, but some of them, and this was the only example by that time in 2016, there was just this one single enzyme that can work at 40 degrees Celsius for degrading PET. And so when we started working on this, there's a Chilean Antarctic Institute that does a lot of biotechnological research. And one of the things that we wondered was whether there were other enzymes that can also do PET degradation at low temperatures. And since the Antarctic regions are so nearby, we were wondering if we can find them over there. And so you will say, oh, but that's, there's not many human activity in the Antarctic areas. Why would you search for an enzyme like that over there? But I'll convince you that it's important and that it might be quite catastrophic to actually find plastics in the Antarctic region. So just two examples, 2 slides. So this is a test that they did with a boat on the Weddell Sea, which is in the Antarctic region. They went around and they were collecting plastics just as much as Carol was presenting in the morning. And they showed that they have a bunch of microplastics in different regions. And if you collect samples from either the surface or from the subsurface, you are going to see that most of them are located on the surface, and they are quite small in terms of size. And most of them, and this is quite different from other examples in the literature in which they find typically like polypropylene or polyethylene, most of them are polyesters. Okay, so first we have some evidence that in some coast in Antarctica, there's a bunch of polyester materials that is being deposited over there. And they're still wondering why this is happening. One option would be that the currents are actually taking that debris over there. The second one was a report that just came out, I think last year, which is, it was sampling these different places in Ross Island, and they showed that they can find different types of microparticles all around the place. So these are the places in which they did all of the sampling. The circles represent how big were the numbers or the concentration of microparticles in each site. And then they did some modeling and they sort of demonstrated, this is present on the snow, by the way. So what they think is what is actually happening is that these are airborne particles that are just floating around, and these are being displaced throughout the whole Antarctic region just by the wind. And then they become deposited in the snow, and then you can collect it. And when they look at with different spectroscopy techniques, they look at the type of polymer that they were accumulating. Most of them was PET. Okay, so PET was found in 79% of all snow samples that they collected, and the concentration is still fairly low. But I mean, it's still shocking that this is so far away from every other continent, and you can still find these particles everywhere. So with that in context, we decided to do the simplest thing that anybody working on biochemistry can do, which is to take a sequence of an enzyme that you know already, put it on a web browser for protein sequences that are similar to it, just as much as you were doing like Googling or Bing, and you will search for similarities for other proteins that have similar patterns, similar sentences, okay? And so this will be Protein Blast. So in this case, we're taking either whatever enzyme that we have here handy, for example, one of the cuttinases that was mentioned by Wolfgang, or the idiolase that was mentioned also in the morning by Beatriz, and then we feed them into this program, and what you get is a result of other proteins that are similar in sequence. And what was interesting is that two of them are actually MORS1 and Odeipira Antarctica glutinase, so two different enzymes that come from Antarctic organisms. They have fairly low sequence identity. They are not so similar to the ones that we know, and they might be potential PET degrading enzymes. And so the easiest way to test for polyesterase activity for a given enzyme is two different things. And both of them are based on the same kind of experiment, which is you take a aliphatic polyester that Wolfgang already mentioned, easy to break, such as polycaprolactone. You make small particles of it, which are called nanoparticles, and you can either put them on an agar plate so that you can actually see that the plate is opaque, and then if there's degradation, you will see a halo of degradation around the bacteria expressing your given protein. That would be one option. Second option is that these particles are not invisible to light. So whenever you put a light source, they will scatter the light, and so the amount of light that you receive on the other end in a recorder is not going to be the total amount that you're putting in. And you can use that as a measure of how fast a degradation process is occurring if you're adding an enzyme that it's degrading the material that is interfering the light. So two different experiments for the same purpose. One of them is qualitative, and the other one is quantitative. So we saw that MORS1 and also the other enzyme, but I'm not showing this in this slide. They both degrade polycaprolactone, and they both show this halo of degradation around the bacteria when they are expressing the enzyme. And when you do the kinetics assay, which is to follow the change in turbidity due to the degradation of the polymers inside this cuvette, you're going to see something like this. So for the two enzymes that we collected, one of them, MORS1, was quite efficient at degrading just in 2 minutes the whole set of nanoparticles that were within the tube, whereas the other enzyme was not highly efficient. And we were using the same enzyme concentrations, and we were using a fairly low temperature, which is 25 degrees Celsius. So obviously, we were wondering whether these enzymes are efficient just at that temperature, or just it so happened that they can degrade plastic at that temperature, but they have a preferred one that is different, maybe higher. So we did an experiment in which we do the same, but we do it at different temperatures. So we spent from 5 degrees to 35 degrees, and you can see that there's a tip, which is the optimal temperature, which is around 25 degrees Celsius. So this enzyme was actually an enzyme that will degrade. It will be initially a polyesterase that works at 25 degrees Celsius. But what about PET? So I'll jump into just right away. If you do it with PET, then you do a different experiment. And the experiment is something like incubating a small piece of PET into a test tube and then adding the enzyme and see either the changing in the weight of the film upon several hours of incubation or following the products of the reaction, which is the terephthalic acid. So if you do that experiment, what you're going to see in the somewhat green bars, it's going to be the weight loss. And what you're going to see in two different shades of pink is going to be the products of the reaction, which were already mentioned before. I don't know how many times I'm going to say Wolfgang in this presentation, but Wolfgang mentioned them in the previous talk, which is the terephthalate and then MHET, so two different products. And so what we see is that if you have a null control in which you don't have any enzyme, then you don't see any degradation whatsoever, which is okay. When you add Idionyla saccharyensis betase, you have some level of degradation. This was already mentioned, again, that the weight loss caused by this enzyme is extremely low. It's not efficient as the thermophilic enzymes. And if we compare it with MORS1, the weight loss for MORS1 is similar to what we see for Idionyla saccharyensis betase, okay? But now the reaction for this one over here is at 25 degrees Celsius, whereas this one is at 40. And when you measure the products of degradation, they're somewhat similar, but not the same. There's a larger amount of MHET that is being accumulated by MORS1, whereas Idionyla saccharyensis betase produces more terephthalic acid. The second thing that will be important for an enzyme to be efficient for degrading plastics is that you can keep using it for a while. So you do an experiment in which you take samples every few hours or days or weeks. And so we did that, and you can see this graph over here that shows the incubation days for looking at degradation. And you can see that the degradation just ever increases, which means that the enzyme keeps working on the substrate, chewing on the plastic little by little. Right, so how it looks like, this is again a comparison of a negative control without enzyme with another one that it's a PET film that is treated with the enzyme for 10 days at 25 degrees Celsius. And these are surface changes that were ascertained by electron microscopy, surface electron microscopy. And you can see that without the enzyme, you don't see any degradation. But when you add the enzyme, you have these cavities on the surface. And again, mesophilic enzymes or psychophilic enzymes are not chewing inside of the amorphous, in this case, plastic films, but they're actually showing just on the surface, which would be like the loose ends of the material that have been degraded and just causing these cavities all over the place. Okay, so the idea for us was not much about applications. At the beginning, it was much more about learning why these enzymes work at low temperatures. And for that, we used two different types of strategies that we typically use in the lab. One of them that we do in collaboration with people either in the US or in Brazil, which is a structural biology, which is trying to get a picture of how a structure of a protein looks like. And the other one will be something that we actually do a lot in the lab, which is molecular dynamics. So simulations in the computer about the motions of a protein. And why we decided to do that? We had a bet, and the bet was that if the plastic becomes more rigid at lower temperatures, the enzyme has to compensate for that somehow. And the way to compensate for that will be for the enzyme to become more flexible for degrading the plastic that became rigid at low temperature. Very easy bet that we wanted to test by molecular dynamics. So we did first the crystallization assays. If you haven't seen how a crystallization assay is, this is your video, very short one. So what you do is that you turn your protein in solution into a crystal by adding compounds that allow for this process to occur. So these are crystals just as a salt, but they have inside just a number of proteins that are all oriented in the same way, so extremely well oriented. And given that you have this mesh of electrons from every different atom, when you shoot X-rays into it, you have a diffraction pattern due to the X-rays clashing with the electrons. So then after that, you get these dots in an image, which after some mathematical calculations, you can go back and convert them into positions of atoms in three-dimensional space, which is what is shown here. So with that, you can get a structure of a protein, and quite easily for some of them. So we got the structure of a one protein, which was Hydrogenase agaryensis petase. By that time, I think we were one of the 5 different groups working on that enzyme and getting the structure, which is shown here. There were some adaptations that were interesting, but we were extremely interested in motions more than anything else. So what we did after getting the structure was to run simulations. And so when we run the simulations, what we do is that we use just molecular mechanics, so Newtonian motions applied to atoms. We tell them that they have to move, they have to wiggle in different places, and they will wiggle more or less depending on their context, whether they are well trapped between other atoms or whether they're highly exposed on the surface, and they will have different motions overall. And then you can, given that this is a computational model, you can quantify how much a given residue in the whole protein moves compared to the other ones. And so the simulations look like this. So remember that the bet was mesophilic enzymes are more flexibility than thermophilic ones for degrading plastics at low temperatures. And so we run a simulation of a thermophilic enzyme and we compare it with the mesophilic. These are the actual molecular dynamics results. So this is a video of a simulation that we run. And we typically run 4 of them at the set temperature conditions at atmospheric pressure that we want, targeting whatever we're doing on the experiment. And so when we run those simulations, you cannot see much of it because they don't move quite a lot. But if you merge all of the images that we collect during the simulation, you will see something like this. So what is in yellow, orange, and purple will be the loops in the active site where the enzyme actually chews on the plastic. And when you have on the right side, it's the same thing, but for the mesophilic enzyme. And you can notice that in the case of the thermophilic one, there's not much movement for any of those loops, any of the yellow, orange, or purple ones. But if you look at the right side, the yellow one looks the same, and the orange one looks the same, but the purple one's moving a lot. And so we can quantify this in the computer, and we can say how much of that motion exists in this protein compared to this one. And this is the blue curve over here, and that's the region that we're looking at. This will be the purple loop. And you can see that if we compare the Hydrogenase agaryensis petase, so a mesophilic enzyme, against 2 thermophilic ones, that loop moves quite significantly more than the other ones. And even if you add the substrate, that movement, that motion is still there. Okay, so our bet was kind of correct. we had an enzyme that could degrade plastics at lower temperatures because it was moving more the active site loops. And so it was not only that, but also there are, as Wolfgang said before, there are modifications in different enzymes that make them actually different in terms of activity, efficiency, optimal temperature. Some of those modifications occur either on the active site or around it. And this is an example of something that happens around it. So what we also know that we knew that from different structures from different groups, the iduronidase agaensis betaise not only had like very flexible loops on the active site, but also there was this little residue over here that is a tryptophan. So tryptophan, you can imagine them as swinging doors because they are very bulky. And so they have to move and rotate in order to allow for something to bind into an enzyme. So if you need to bind something here, like this molecule over here, which is an analog of a substrate of a plastic, this thing has to move around a little bit. Okay, so when it moves, it finds a specific conformation, then the substrate can bind into it. And so in the case of iduronidase agaensis, it was described that there were two residues away from the active site, but that they were allowing for this change in position of this swinging door within the enzyme. And so if you compare it with other enzymes, they had other residues that were impeding that movement. And the hypothesis was that if you reverse the mutation in iduronidase agaensis betaise by those that you find in the other thermophilic or mesophilic enzymes, you will have a decrease in activity because now the swinging door is locked into a position. It's not moving around. And you can see that in these experiments that were published in 2021 from a different group, in which they see that whenever they add the double mutants, they replace these two residues by these other bulky ones, you don't have any activity. And if you do the reverse, if you add these highly flexible residues and you allow the swinging door to move in the thermophilic enzymes, then you have an increase in activity for the thermophilic ones. And it was said that this was a generalized method for editing, engineering an enzyme to have better activity. Again, we knew that that was not only because of the identities of the residues, but we knew that this was because of an increase in flexibility. And here you see the results. In the same way that it was done in the experiments, if we reverse the mutations in the iduronidase agaensis betaise in the computer and we run simulations, there's no flexibility after doing the mutants. And if we do the reverse mutation on the thermophilic enzymes or mesophilic ones, we have an increase in flexibility. And what you have here, this shading region, it's the statistical deviation of the results. So this is consistent if you run many, many, many simulations. So we knew that dynamics and this wobbling of the tryptophan were important for the activity. And therefore, given that there are so many publications about how to engineer these enzymes and make them better, we tried all mutants that were published before in our enzyme. I'm gonna skip into that here. There we go. And so when we did that, we tried different mutations. So we tried that one that I just mentioned, which is a double mutant. We put it into an enzyme and we tried to test for how much activity we have after doing that mutation. And we were expecting for our enzyme to be better, but it's not. It actually loses most of its activity. Then if we try all different mutations that have been published so far for all of these different enzymes, none of them actually have more activity. All of them are worse. And we were puzzled by this. So what we did, instead of doing changes mutation by mutation, residue by residue, we decided to go for something more aggressive, which is what if we change a whole piece of an enzyme? And so what we decided to do is that we took this highly flexible loop that we saw before. We said, what if we change it for the thermophilic one? So we have a mesophilic enzyme, degrades at 25 degrees Celsius. What if we put something else in there? And in order to try to increase the activity. So we put it in, and we took a piece of LCC, one of the most well-known thermophilic enzymes. We put it in there, we replaced it. And when we did it, there was no activity. And the reason that we had no activity is because there's another interesting fact about these enzymes is that they have this connector over here in yellow, which is a covalent bond. This is present in mesophilic enzymes. This is also present in psychrophilic, but it's not present in thermophilic. And so we said, maybe we have to put it back. And so when we did it, when we put it back, we have an increase in activity we recovered the activity of the enzyme. So we had a mutant that is a replacement of a loop. We put back whatever the enzyme needed, and now we have activity. And this is a message maybe for people doing biochemistry and protein engineering that are trying to solve issues with the enzymes that degrade plastics, is that there's no change in activity that doesn't come with a potential change in thermal, in the optimal temperature for activity. And this is what we saw here. We tested again now with PET films whether the optimal temperature for activity was the same, 25 degrees Celsius. But when we did the change, when we tried to improve this enzyme by this engineering process, it is not. Now we have a shift of 20 degrees Celsius, and the enzyme degrades PET at 45. So the modification that we're doing is not only altering the activity, but it's also altering the stability. And the other thing that we did was to compare just the wild type enzyme with this modified version. At 25 degrees Celsius, they're somewhat the same in terms of weight loss and in terms of the accumulation of the different products of degradation. But if you were testing for the activity at 45 degrees Celsius, which is the new optimal temperature, then you're going to see that this enzyme is 4 times more efficient than the previous one. And these are reactions that took, if I'm not mistaken, just an hour. And so we can obtain increases in activity for these enzymes, but some of them might lead to these changes in optimal temperature that we might not want to have. And why am I saying that? And this is the last few slides that I'm going to use for this. Wolfgang already mentioned that there's already like an industrial concept or proof of concept of using of recycling PET by using enzymes, which is this company that is over here. Now, in this case, they do want a highly efficient thermophilic enzyme for degrading plastic to convert it, hopefully, to 100% terephthalate so that they can then produce more plastic afterwards. But that's because those are countries with plastic producing capabilities. Chile doesn't produce PET. So what do we do? And so we saw already that you can use the PET enzymes for the PET degrading enzymes for modifying surfaces. You can use them, and it's a great suggestion from the previous talk that we can actually try to avoid dumping clothing that it's made of polyester by trying to modify the surface and get rid of the peeling by using these enzymes. But what about recycling? What will make sense for a country like us in which we don't produce new polymers? And so my very humble suggestion will be that maybe we have to do something more ambitious, more complicated, and that it will take longer, which is something that is already happening. So there are some people that are already working on this, which is using organisms that can take the terephthalate and that can transform it into other high value compounds. And these are just 2 examples. One of them is from 2021, and the other one is just recent, just from January this year, in which in one of them they use a PET and a process that it's called upcycling, because the bacteria takes the terephthalate and produces something else. They produce vanillin. And so vanillin is used for food. I don't know if somebody will eat that, but this is highly, highly expensive. And so there are ways of doing chemically, but this will be an option for doing the same process using recovering PET and then using it as an entry product for making vanillin. And then another option will be to produce, in this case, they produce lycopene, which is a carotenoid that is used, so it's present in tomatoes, that's why they are red, but it can be used for getting, for example, you can use carotenoids for developing pharmaceuticals or even getting other types of high value compounds. So this might be options in a country like us, in which we don't have the PET producing capabilities, and probably we are not going to be able to sell very expensive TPA from our country to other countries. This might be a more suitable option for a country like us. So with that, I would just like to thank, this is the group of people that are from my lab. They are in the back, so you can say hi. Some of them, not all of them, are working on plastic degrading enzymes. We work on protein evolution overall. But so the group that is working heavily on plastic degradation would be Aranza. We had Amparo working on this team before. Felipe Gatica has been working on it quite a lot. And then Pablo was working on it, but it takes for a while. But I would say that the starts of the things that I said today were Paula, who's now at Leipzig University, and Gokhan already mentioned her, and also Felipe, that is also at Leipzig University, just in a different group, working on computational stuff. And then we do have collaborations. Like I said, I think none of this research would have been possible if we were not having Wolfgang with us, working remote on these very crazy ideas. We collaborated with people doing structural biology, as I mentioned before, from Brazil and from Ohio State, Richard Garrett and Marco Sotomayor, with whom we have gotten most of the structures that we have now. And with Beatriz, with whom we've been looking for enzymes in Antarctic environments more generally. And then a lot of funding. So one of them is actually the funding that we're using for this symposium, which is a joint grant between our national institution for research and the Foundation for Research in Sao Paulo, the Millennium Institute for Integrative Biology, which is also chipping in for this symposium, and the Chilean Antarctic Institute that gave both Paula and myself grant for research on Antarctic enzymes. Thank you so much. Does anybody have a question? So otherwise I'll jump right away into the other talk. Okay, Wolfgang has one. Yes. So I will dare to say, without much knowledge, that degrading the terephthalate might be putting it at a selling point that might be too expensive to sell it, and so perhaps other countries wouldn't buy it because we will have to sell it to somebody. So I'm not sure if we can make a profitable industry of recycling out of it. That will be my very honest question, but I don't really know.
Yeah, I mean, there's a market for terephthalic acid, and everybody who want to make a PT would need to buy it. And if you have a competitive price, I mean, it has to be lower than the oil made.
Right. That will be the challenge, I would say.
But if you consider how much it will cost to produce the vanillin, I wonder if you can make a profit.
Yeah. No, I do so too. So I think that those are just early experiments of what we can do by combining the products of degradation with upcycling, but it's not fully explored. So I do believe that it's an opportunity for doing something, but it might go nowhere. That is true.
Well, you have to look at the market and how much cost is further involved and who would buy it. No, you have to research that.
Yeah. Thank you. I think Gabriel has a question.
Hi, Cesar.
Hey. Yeah, go.
Thank you for your talk. That was very interesting. My question is, in order to you, what's the biggest difficulty or the barrier to studying microplastic in this country?
Oof. We were talking with that a little bit with Cristobal, and I think some of the challenges are more related to how to collect microplastics in a very large country, and the fact that we're so little, with such a small group for understanding that we don't have the capabilities of doing some of the things. Well, not the capabilities, but I think we don't have the drive yet for doing what it's been done in the Northern Hemisphere. There's more incentives. I think that the incentives for research, microplastics or plastic degradation or upcycling are taking place now due to the new legislations that are taking place in the country. But after legislation, then to put forward like different industries, like emerging industries with biotechnology purposes on their backs, still takes a while. So I think that my honest answer will be that if you compare us to the Northern Hemisphere, we are kind of in a delay, and that our advances in research in these areas will take a few years. Yeah. All right, we're good? Oh, small? Okay.
Thank you. Actually, this is something I always wanted to ask a specialist. So when you're engineering a protein for example, thermal stability, how much of it is rational design and how much of it is basically just trying out different mutations and seeing what it does? Because I've been reading a lot of publications and it seems to be both seem to be strategies that are employed, but a lot of it also seems to be just luck or trying out.
Yeah, I think there I'm in line with what Wolfgang said before. And this was said not only by Wolfgang, but many people working on protein engineering, including Francis Arnold, which is much better to start with a highly thermostable enzyme than to work with a mesophilic one and then try to make it thermostable. Just because the thermostable scaffold is resilient to every type of mutation that you can do. So you can test, it's much easier to test for changes in activity without compromising its ability than to try to edit stability afterwards. And so if you look at most people that are working on protein engineering, they prefer thermophilic scaffolds to begin with. And some of, I don't know, I can just mention, again, in the case of Frances Arnold, she works with an enzyme that is like cytochrome P450 that she uses for almost every work that she has. And those are all thermophilic hyperstable enzymes. Yeah. So you can do it now with, just to add to that question or to that answer would be that now with the advent of artificial intelligence, maybe you can do a lot more. And there are some examples of it. So the Idaneosargensis species was edited by using artificial intelligence for increasing a little bit their thermal stability, but most successes with thermal stability have been linked to directed evolution, I would say.
The second question I have is actually both to you and Wolfgang, if he also wants to contribute.
Sure.
I was wondering, so he also said in his talk, and we also see that actually the enzymes that we isolate as plastic degrading enzymes are not plastic degrading enzymes. So this is not the main gig of this microorganism, because obviously the enzymes haven't had the chance to evolve to degrade plastics yet specifically. My question is, do you think this is ever going to happen? Is plastic a driver of enzyme evolution in the nature? Because I mean, imagine how long it took after the Cambrian explosion for first lignin-degrading enzymes to emerge, just like many, many thousands of years. So, do you think this will ever happen, that we will have really specific plastic-degrading enzymes and really organisms which specialize in this?
I'm doubtful.
Okay.
I would say that I'm doubtful. We were just talking about this the other day with Wolfgang, which is that there are organisms that consume terephthalate without having the capabilities of degrading PET. And then you can conceive that what will be an organism that is adapted to eat plastics? It will be like a highly efficient one that just survives in plastic, probably. But it might be that the capabilities for degrading the terephthalate or metabolizing terephthalate are already there. And then you have an enzyme that just happens to be efficient at degrading it without plastic being a driver for that selection process.
I have to add though, there are terephthalate degrading enzymes, but there are not really that many of them. Like I think I know like 2 different pathways which are degrading terephthalate. I mean, if you imagine how long it has been around, it's not really something. So I think there was like one evolution and then it just got spread out. So it can actually, even that is not that common.
Yeah, but then I would say that also the amount of evidence that you will need to build up to prove that will be quite a challenge. Yeah. So I don't know if you want to add something onto that.
I agree. It's maybe it's not even necessary because it may be that biology is not the only way we get rid of this stuff eventually. It may just take a long time. Like how we get from the macroplastic to the microplastic, it works just with sunlight and mechanical activity, for example, of the waves, which breaks it down further and further. And then small parts of the molecule may be exposed, and this is readily degraded. like terephthalic acid, it will not stay in the environment forever. It's degraded somehow down to CO2, maybe not very efficient, and you cannot use that in industrial process. But on the long term, it will all be gone. And if not, it will kind of sediment into the floor, sea floor, and it will become the, what they call it, the plasticine. Right, right. Already has been indications that this will happen, a geological feature in the future.
Thank you. All right. We move on to the last talk of today's symposium. So I'm glad that Başak is going to be the next speaker. So she's coming from Bilkent University in Turkey, but she's been working in Germany for quite a while. And she's working on the biodegradation of biodegradable plastics in marine environment, with a lot of interest in combining many of the things that we've seen today, which is metagenomics, microbiology, environmental microbiology, plastic degradation, and so on and so forth. So without taking much time, I would like to thank Başak again for traveling to our beautiful country to give this presentation. Thank you so much.
Well, actually, it is my turn to thank you for the invitation and this amazing chance to talk about the work that we have been doing in the past year. So I'm going to talk about something a little different. I now notice that my title is a little chunky, but actually what I'm going to talk about is biodegradable plastics. What are they? Why are they actually a thing? And their potential fate and biodegradation by marine microorganisms. I'm going to focus on a specific type of biodegradable plastic, which are the aliphatic and aromatic ones. Actually, Wolfgang briefly showed them in his talk, but I'm going to go a little bit more into detail and talk to you about their potential degradation and the mechanisms thereof. I guess I don't have to talk 15 more minutes about this, so we can just accept this and move on. I will have to say, though, the Alfred Wegener Institute has a very nice website on marine litter with a lot of nice numbers from all over the world that they've been gathering really interesting data for a very long time. So if you would like, so if you're still doubting that this is a problem, you can actually go and check their website. You don't have to take my word for it. Biodegradable plastics have been devised actually already a while ago as one of the potential solutions to combating the plastic pollution problem, not specifically to the marine environment, of course. The major difference between biodegradable plastics and the durable plastics is that biodegradable plastics have biodegradable backbones. So these are typically polyester bonds, so CO, rather than the carbon-carbon bonds that typical polyolefins such as polyethylene and polypropylene have, which are more susceptible for enzymatic degradation. Here are two different kinds of example biodegradable plastics. The polylactic acid, for example, is composed entirely of aliphatic monomers. And the polybutylene adipate terephthalate, which I'm going to talk more about today, has an aromatic component and also an aliphatic component. These polymers have some other properties which make them biodegradable. For example, they have a looser crystal structure compared to other plastics. They absorb liquids more readily, so the enzymes have a chance to access them better, and also some other properties. But being made from renewable resources is not one of these properties. So I think we should really take a moment to clarify this. There is much controversy about biodegradable plastics, and I think one of the reasons is that these are often put under the umbrella term of bioplastics, which mean both biodegradable plastics and bio-based plastics. Biodegradable plastics can be made from renewable or fossil resources. For example, the compound that I just mentioned, polybutylene adipate terephthalate, but also polycaprolactone that Cesar was mentioning. These are made from fossil-based monomers, and they are biodegradable. On the other hand, you can make polyethylene or polyethylene terephthalate just from sugar, and this will not be degradable. There are also biodegradable and bio-based plastics. For example, PLA is one of them. There's also PBS. Even starch can be made to be some sort of plastic, and it can also be blended with other compounds. Where the monomers come from have nothing to do with if this plastic is biodegradable or not. It is only the final chemical structure which determines biodegradability. And this is why I don't really like the bioplastic term, because then people say, oh, but you said it's a bioplastic and it didn't degrade, but it could be that it was not really meant to degrade in the first place. The applications for biodegradable plastics, first of all, I'm aware, and you guys might also guess, that biodegradable plastics will never completely replace all plastic products. It's also not the idea. A lot of plastics are made to be durable, and this is a good thing because we can reuse them, which is what we should be aiming to do anyway. Obviously, you don't want to lay water pipes and then have to change them after 6 months because they have corroded or degraded, but there are quite a few applications where either the plastic is short-lived or it has a high potential for loss. We saw this morning that just due to the use of the plastic, for example, for car tires, for the soles of your shoes, for fishing nets, there can be some loss which we cannot prevent. As it was mentioned in the other talk, there are some products which are just manufactured in a way which are not very good for recycling. For example, imagine the paper cup that you use for hot drinks. The paper is recyclable, the plastic is recyclable, but if you then coat your paper cup with the plastic, it becomes just, you can only incinerate it basically, you can't do anything. But if the plastic coating is biodegradable, there's a chance that you can put it in an industrial composter and it will be completely composted and you will not generate any, for example, microplastic or any other residues. It is the same with compost collection bags, coffee capsules. I hate them, but you can make them out of compostable plastics, which means that, again, you can just put them in a composter and they will be completely degraded. Another application which I didn't show here is, for example, agricultural mulch films, which, again, you cannot really recover them very easily, and it's nicer to have a product which will then, after a certain amount of time, degrade. Biodegradability is complicated. So I know biodegradable plastics get a lot of bad reputation. You might have seen news headlines, oh, but you know, I put this in the water for 6 months and I took it out and it didn't degrade, which means it's not biodegradable. Okay. Biodegradable plastics are biodegradable. They are certified using international standard methods. And the certification schemes are very rigorous. You just have to look at what the biodegradable on the label means. And this is actually usually quite well clarified. In most cases, biodegradable will mean compostable. So if you put it in an industrial composter, I don't know, in Turkey, this is not really a thing where I come from. Like, we don't really have industrial composting facilities, or they're not widespread. And we don't collect this kind of waste separately. So if you're living in a country which these exist and there's this infrastructure, it's great. Otherwise, yeah, this is a little bit meaningless. But even in countries where this happens, sometimes companies don't want to have these in their composters because they think their customers don't want it. I don't know. But they are biodegradable. You just have to look at the certification. For example, this label is for plastics, which will biodegrade in soil, which means that they have been certified for soil biodegradability. This is quite common in agricultural mulch films, for example. And if you have a little seedling sign here, for example, this means that you can put it in your home composter, so you can compost this in your garden. The biodegradable plastic industry often is blamed because of greenwashing, and there is some greenwashing going on. For example, if you buy such a plastic bag in, okay, you can also see it's very green, you know, it's advertised as a green product. If you buy something like this in Turkey, it says it is something, I think if you translate it literally, it means like it's dissolvable in nature, which gives you the impression that It's basically like toilet paper. If you put it in water, it will just go away, which is really misleading. But as I said, the testing is done, and normally these plastics should be degradable for the things that they are certified for. The problem is, if we talk about the aquatic environment, there aren't really reliable testing schemes for certification for biodegradation in water. So if someone says, yeah, but you said it's degradable and it didn't degrade in water, then you can say, okay, but I never claimed that it will degrade in water. At the moment, nobody can claim it because there is no such certification scheme. I do have to add, though, this is nothing special to plastics. Biodegradability of anything is determined by environmental parameters. For example, human beings are very degradable, but under unsuitable conditions, low oxygen, low pH, we will also be conserved into a bog body. Of course, there are some things which are inherently more degradable than others, but if you're talking about degradability of anything, you have to say what environment and what kind of conditions you're talking about. And this is also the same for biodegradable plastics. So what is the problem with the aquatic ecosystem? So why don't we have a nice certification scheme, just like the other environments? Yeah, well, the ocean doesn't look like a bathtub, obviously. If you think about the structure of the marine ecosystem, you have so many different environments, basically. So marine is a very broad umbrella term, which means a lot of, basically it's like a compilation of all these different zones. You have the coastal ecosystem, it's higher in nutrients, maybe higher in oxygen, lots of biomass. You have the pelagic water column with no nutrients and very little biomass, where any activity is not going to be like very much. You have the benthic zone, the water sediment interface, again different. You have, God forbid, the deep sea. So a friend of mine is a deep sea microbiologist, and he told me that just to see what happens, he dropped an apple like 2 kilometers down during a submersible drive, and 2 years later it was still there and completely intact. So obviously, if your plastic particle sinks into the deep sea, you can't expect it to behave in any different way. So yeah, marine zones actually differ in terms of their redox environment, their accessibility to light, how much biomass is there. And this is very difficult to accurately represent under laboratory conditions in a standard testing scheme, which has to be fast, it has to be cheap, and it has to be performable in all kinds of different labs. So when we started the research, basically, we wanted to ask, first of all, the question, can marine microorganisms degrade these biodegradable plastics at all? Because the data that was available was only from terrestrial organisms which don't live in the marine environment. If so, which microorganisms do the degradation? What are the mechanisms? So how do they degrade it? And which environmental parameters play a role in the biodegradation of these plastics? So I'm going to talk a little bit about the cultivation and characterization of such plastic degrading consortia, how we elucidated the biodegradation pathways, how we characterize the plastic degrading enzymes, and also then I will take a little step back and maybe talk a little bit about the biodegradation of plastics in the natural environment. But this will be like a very small part because it's a project which is still running. I have a confession to make. I started this project out of spite. So basically, I started working in the chemical industry some 5-6 years ago. And I was working in a very big company as a research scientist they gave me the task to investigate the degradation of these plastics in the marine environment because they were getting a lot of pressure from NGOs, for example, governments. Okay, what happens if your plastic falls into the sea? And I had a meeting and somebody told me, yeah, but it won't degrade anyways. And I had another meeting and they said, yeah, it's not going to degrade. And the third one said, yeah, but you're working for nothing, it's not. And at that time, something clicked in me and I said, okay, I'm going to make this degrade because just so many people told me it's not possible. And I know it is possible. It must be somehow possible. You know, there are microorganisms who do this. And how we went about this is basically very easy. It's good old microbiology. There is no secret. And I'm hoping after my talk in 2 or 3 years, also in Chile, there'll be a lot of papers about microorganisms which can degrade this and that biodegradable plastic, because I think really everybody should do this. It's really nice and very rewarding. All you need is a marine sample. Here, I think I should change this image, because we didn't take samples from plastic litter or anything. We just went to the beach and took a handful of sand, a little bit of water, and this was our inoculum to grow these microorganisms. You also don't need a shark. And afterwards, okay, to get the initial degradation is a little bit more difficult. I tried in my Friday afternoon project to begin with a lot of different conditions to increase the biomass, to maybe promote the growth of microorganisms which like to attach to surfaces and such. And our carbon source, in this case, it was a commercial blend of polybutylene adipate terephthalate. I wanted to work with a commercial product because I wanted my research to be relevant. It was a big mistake because they didn't tell me what it was for the longest time, and I didn't know what I was working with. And this is a big problem if you're working with biodegradable plastics. These are not polyolefins. They are very, very strictly protected by intellectual property. They usually have some brand names, but if you buy one, you will never know what it is. Nobody will tell you. Even within the company, not everybody knows what everything is. So this is one challenge of working with this kind of compound. But basically, I inoculated my flask, and afterwards I waited for some time, and after the first degradation occurs, so it's actually nice, you can see that it's degrading, it just disappears. I inoculated this into a fresh flask, this time with no other carbon source than plastic. And after I did this transfer several times, I saw that the consortium, the enrichment culture, was stably degrading this plastic. I did a little systems biology experiment where I investigated the metagenomes, but also metatranscriptomes and metaproteomes of this consortium to see who was out there and how the degradation was happening. By the way, I'm going to give another talk, I don't know when yet, I think towards the end of this month, about the metaomics techniques themselves. So more going into depth about how we do these things. So if you are more interested, maybe come to that talk, because I'm not going to go too much into detail here. So first of all, I was right, and this is what matters. The marine bacteria can degrade polybutylene adipate terephthalate very fast, actually. So here you can see that with our consortium at room temperature, after 3 days already, we saw that there are some holes and cracks on the surface of the plastic. You can actually see that the surface is very densely colonized. And already after 6 days, you can see bigger holes and larger craters. And in these craters, you can see the biofilm is nicely nestling. At this point, the plastic is really like toilet paper. It's basically disintegrating. And within about 20 days, you can see the complete transformation of this plastic into CO2 and biomass. Just as a reference, in an industrial composter, much higher temperature, much higher biomass, this takes 6 to 8 weeks. So our bacteria, even at ambient temperature, can perform this a lot quicker. So the setup for the omics experiments was a little bit complicated. So normally, if you're doing a comparative approach, for example, you want to see how a certain pesticide is degraded by a certain organism, you feed it, for example, glucose, and then the pesticide, and then you compare the gene expression when the organism is feeding on these different substrates. If you have a consortium, you cannot really do this, because if you add another substrate, this is completely going to shift the composition of your microbial community. So instead, what we did was we grew the microorganisms on the plastic for 30 days, and then we gave them a 1 week of starvation period to really bring down the gene expression. And afterwards, we added fresh plastic, and then we did a time series to track gene expression profiles from the first edition until the end of this time period where the plastic was completely degraded. In addition to this, we also analyzed the biofilm community which was basically growing on the plastic film, and the free living community, which was growing in the culture medium separately, just to see if these are different and if there are organisms specialized to live on the film and in the free living fraction. What we saw that, first of all, although we did quite a lot of selection, our community was still quite diverse. And just like Beatrice was telling in the morning, microorganisms sometimes need each other, so they can't really grow as well if you pick them into single pure cultures. And I also never managed to isolate a single bacterium which could degrade this plastic, but within the consortium this worked really well. The structure of the consortium was quite diverse. We had 33 different species, which we determined by, again, the method that she was mentioning. So we assembled genomes from these metagenomes, and we assigned them to certain species. We saw that there are 33 different species growing. Not all of them are very abundant, but they, if you look at the bubble plots, the community structure was quite stable throughout the degradation, so we didn't really see major shifts. But if you look at the biofilm versus the free living community, you can see some differences. For example, we had one strain, a Marinobacter strain, which was much more abundant in the biofilm than in the free living fraction. And we had another bacterium, Pseudocyanicola, which is an alpha proteobacterium, which was a lot more abundant in the free living fraction than it was in the film. So there seems to be some sort of specialization, but we didn't find any exclusive species, which probably means that the attachment and detachment process is quite dynamic and it's ongoing all the time. The differences were also confirmed in the omics experiment. So if you look at the gene expression profiles, the T, we saw that, so the red is the biofilm community and the blue is the free living community. You can actually see that our Marinobacter was a lot more active in the biofilm, along with another few organisms. And our alpha proteobacterial strains were a lot more active in the free living fraction. And also the protein production also showed us a similar picture. So the Marinobacter was responsible for most of the protein producing activity on the biofilm. And our Pseudocyanicola, which was also more abundant, produced most of the proteins which were upregulated in the free living fraction. So there is some sort of labor division within this microbial community, which is quite evident also in the systems biology experiments. When it comes to the elucidation of the degradation pathways, we refer to, first of all, the existing literature. Most of the existing literature which is out there on the degradation of aromatic polyesters is actually from the degradation of polyethylene terephthalate, which I'm so glad has already been covered many times, so I will not go very much into detail here. Have a little look. Okay. So, but why are we talking about PET now? Because actually the compound that we are investigating here, if you look at the structure, it is quite similar to PET. So it has this aliphatic tail, which is composed of basically a dicarboxylic acid and a diol. This part is very easily degraded, so it just disappears, basically. Afterwards, you're left with this aromatic moiety, which is very similar to the MHET that was mentioned before, except that the diol is a 1,4-butane diol rather than an ethylene glycol, which is present in the PET. It's just a little bit longer, but structurally it is, again, quite similar. And we, in our metagenomes, we found 6 putative PETases, so enzymes which look like PET-degrading enzymes which are isolated before, and we hypothesized that these are degrading the polymer into this intermediate. And we found another enzyme which was related to the MHET degrading enzymes which were found before. And we decided that it is likely that this second step is performed by this secondary enzyme, which then produces terephthalic acid. Again, we found terephthalic acid degradation pathways. These are quite conserved. I was actually very surprised, but even in the marine environment, they are very closely related to those from the terrestrial ecosystem. But yeah, so after we got our candidates, I'm going to walk you through this. It looks complicated, but it's not. We developed a putative degradation pathway. We saw that two PETases, so putative PETases from Marinobacter, are a very active bacterium. They're very highly upregulated in the biofilm, so these we called 628 and 453. These have secretion signals, so they are secreted outside the cell, which makes sense because you cannot really take the plastic into the cell, so you put your enzymes out, and they degrade it, and then you can use the smaller molecules as a carbon source. The Marinobacter doesn't have any enzymes for degrading terephthalate, but it does grow really well on the diols and also especially the dicarboxylic acids. So it degrades the polymer and it feeds on the aliphatic parts of it. Then what happens to the monomers? Basically, the aliphatic parts, as I said, get degraded really quickly. The intermediate is degraded by a secondary enzyme, as I was telling before. This is very highly upregulated in the free-living fraction. It does not belong to the Marinobacter. We don't know who it belongs to, but it is encoded on a mobile genetic element, which means that it can be transferred from bacterium to bacterium, which is actually good news, which means that these degradation capabilities can spread by horizontal gene transfer. And after this is degraded into its smaller components, so terephthalate and butanediol, again, two different organisms, another gamma proteobacterium and also another one which is, again, encoded on a plasmid. So these have the necessary aromatic degradation pathways to then convert terephthalate completely into CO2 and carbon dioxide. So you need 1, 2, 3, 4 different bacteria to completely degrade this plastic into its smaller components and then completely to CO2. Now it is still a cultivation-based approach, although it's not completely cultivation-based. But I think this is not an unreasonable hypothesis of how the degradation could be happening in nature. So a lot of microbes come together and each feed off different bits of this plastic. And this was our proposed degradation pathway, but it is still only bioinformatic evidence. So we don't actually know if any of these enzymes are a thing. So are they really enzymes? Do they do anything? And there's unfortunately no other way to do this than isolate these enzymes and do activity testing. And this is also what we did. For the putative PET degrading enzymes, so PETases, we had two candidates, the 628, which I mentioned before, and 629. These are both located on the Marinobacter genome. We saw that the 628 was highly upregulated during degradation, but 628 not really, so we could actually hardly detect the protein. These exist as a tandem pair. Okay, this is actually really interesting. So 628 and 629 are on the same open reading frame, so one gene after the other. There's no other open reading frame in between. They don't get produced together, so there's a transcription termination site in between them. They are very closely related to each other, so 73% amino acid identity. And I have to say, this phenomenon occurs really frequently if you look at different genomes where such PET degrading organisms have been sequenced. You can see it in Clostridium, you can see it in actinomycetes, you can see it in even different Marinobacters. Why? Why do we have two different enzymes if they are so closely related to each other? And these are also very closely related to non-PETases, especially the one from Ideanola, which was mentioned before, which is not a surprise because these enzymes are quite conserved. Our questions were, first of all, what are their biochemical characteristics? Are there any differences between these two enzymes? Do you really need both of them for the degradation? Do you just need one, since only one of them was upregulated. So what is going on? First, we elucidated the crystal structures, which look very pretty. They didn't reveal any surprises, so just by looking at their structures, you couldn't tell an immediate difference. The only difference is that the 629 has an extra loop. We tried to engineer this out, but it didn't really change the activity very much, so I don't really think this is a determining factor. The catalytic triads, the lipase signatures, they are exactly the same as other known PETases as well. So yeah, very conserved structural backbone here. If you look at the activity though, there's a surprise here. So we tried the activity testing on 4 different plastics. So the PB80 with the adipic acid as a dicarboxylic acid, PBSET, which has sebacic acid, which is longer as a dicarboxylic acid, but these are just derivatives of one another, so related plastics. The blend of these two, and also polyethylene terephthalate nanoparticles. Now, we saw that sorry about the formatting, something might have gone wrong. We saw that the 629 was actually a lot more active than 628, although this was actually not even produced, the production of this was so much less than this enzyme, which is so much less active, basically. In the case of the biodegradable plastics, the main degradation product was this monoester, so basically the counterpart of MHET. But in case of polyethylene terephthalate, the enzymes were actually quite actively and efficiently degrading the intermediate into terephthalate. So the intermediate didn't accumulate as much when using the not biodegradable plastic compared to using the biodegradable plastic. Probably the intermediate product of the degradation of the biodegradable plastics has some sort of inhibitory effect, which we could also show. And it doesn't fit very well in the active site of the enzyme. We don't really know why at the moment. But although these enzymes are very similar, they produce the same degradation products, and they have different activities, and we don't really know why yet. When it comes to the second intermediate enzyme, the MHET-like enzyme, we can actually see that it does what it says on the tin, so it degrades the degradation intermediate, so the monoester quite rapidly. It doesn't degrade the diester, so the BHET is not degraded. It's very specialized in just degrading the end degradation product. And these two enzymes, or the combinations thereof, can then work together when it comes to the complete degradation. We even did a little, I didn't want to do this, but I had a really enthusiastic master student, and she came to me and said, I have read all these PETase papers, and they're doing such cool evolution experiments, and I also want to do it. And I said, okay, why not? We're using another improved Ideonella PETase as a template. We actually managed to even increase the heat stability of this enzyme a little bit by 10 degrees by doing 6 amino acid mutations. It is not really my main gig, so I don't really work in this field very much, but I actually quite enjoyed seeing that the literature that has been out there until now, where people have done this work, can also be applied to other enzymes, although Cesar says they can't. I don't know. In our case, it worked, but you have to try out different things. So you can actually improve them even a little bit. Probably they will never be like a very biotechnologically important one, but maybe they will. I don't know. I just wanted to show that it is actually possible to use existing information to also manipulate novel enzymes, just because of their similarity. With this, I actually quickly want to conclude by saying that consortia are not very well studied in terms of biodegradation. like microbes working together, but I think there is really a lot of potential also maybe for biotechnological applications such as plastic waste treatment. Why not? Because they just grow so much better together, like even in a nature-like environment. We have seen that although the microorganisms themselves are very different phylogenetically to the terrestrial microorganisms, which we know that degrade these compounds, the enzymes are quite conserved, and they are very similar. And the synergy between them enables complete degradation. I am still baffled by this, but I think I have an explanation which also ties into what Wolfgang was saying before. So we saw that the expression of the genes and the activity of the proteins don't match together. So we see that this enzyme, which is not active, is being produced a lot, and the other one not really. I think this is really because these enzymes were never really meant for the degradation of this kind of compound. So in nature, they do something else. And the transcription mechanism, so the activity of the promoters, for example, are not really tuned to this kind of compound. So they're just being produced in a way that the microorganism is used to. But the adaptation has not really occurred to really fine-tune this expression to this new kind of compound. And this is probably why we see this kind of discrepancy. At the moment, I can't really tell you. There is no real method to measure what happens in vivo, so inside the cell, which enzyme contributes more to total degradation, because if one is not produced very much and the other one is really produced a lot, although one is not very active, it could actually be doing most of the degradation, so we don't really know this yet. At the moment, my main questions are, first of all, how does the biofilm formation work? So how do microorganisms detect these surfaces as something profitable, nice to form biofilms on? Because biofilm formation is the first stage of biodegradation. Your plastic surface has to be favorable to be colonized by microorganisms. In other cases, people work against this, so they make antifouling surfaces. They don't want bacteria to grow, but in this case, you do want them to grow. So this is very important. We would like to find out how this happens. In terms of the gene expression regulation, I was telling you, I think maybe there's some potential being lost, maybe just because these microorganisms are not producing the right enzymes. They are not using their biodegradation capacity fully. Maybe if we can find out what activates this kind of promoter and think about incorporating this either in the culture medium or into the plastic itself, we can actually improve the overall biodegradation. But first of all, we need to find out what makes this kind of promoter tick, and we don't really know this yet. The next part is the development of better standard methods for biodegradability testing in the aquatic environment. So this is a little bit controversial because people say, yeah, but if you certify these things as biodegradable, then people will just throw them away and they will not really think about it. I don't know. In many cases, industries, so the ones that I have worked in, are thinking about keeping this business to business. So basically, you will tell your business partner that this is biodegradable, but you will not tell your customers. I don't know if this will work because these materials are costlier than just regular plastics. And if you don't market them as such, I don't know why people would be willing to pay more in this case. But developing better standard methods is important nonetheless, because I think so people often ask me, why are you working with biodegradable plastics and not, the important ones, basically. One reason is because I like to get results, and it's just so much faster and quicker, and you can even find really cool enzymes in a shorter time. But honestly, I think there's really a lot at stake here, because if we know how the degradation occurs and what are the mechanisms and how we can make things and how we can test this, maybe we can already think about the biodegradability in the material design process. So we can design these plastics and engineer them already with the biodegradability in mind. This is not really the case so much at the moment. Even in the biodegradable plastic industry, they think of all kinds of applications. I don't know, the oxygen barriers, are they a good oxygen barrier? Do they have a good plasticity? Do they have a good processability? Do they retain moisture or not? And so biodegradability, even in this case, comes at the 6, 7, 8 priority level. But yeah, I think maybe in some cases, if we know that there's a high chance that this might end up in an environment that it's not intended for, maybe we can really think of making materials more biodegradable. But first of all, we need to know what makes them degradable and how we can test this. So in terms of environmental biodegradation, at the moment, there's an ongoing lab test where plastic films are incubated at a lake. So this is freshwater, not marine. And samples are taken every 2 months. And the degradation is assessed visually. And we do a microbial community analysis starting from 16S, so just phylogenetic analysis onto metatranscriptomics, so gene expression analysis of both the plastics and the surrounding environment. And we also do accompanying cultivation studies. So our aim is to tie field tests back into the laboratory and thus close the loop, hopefully. This is still ongoing. We have finished taking the samples. Now we have a lot of data. These still need to be analyzed. Good. With this, I would actually like to thank my collaborators and group members. Our funding agencies, we are happy to be funded both by industry and public sector. And if there are any questions, I'm happy to receive that, although I know it's late.
That was a late thank you, but thank you so much, Başak. So Mario is going to go first.
Thank you. Thank you for the talk, very nice. I have some comment and a couple of questions about the biofilms. I feel that biofilm is just a stochastic event in relation to the hydrophobicity of the bacteria or the attachment capabilities according to the surface you provide. So if you have strongly hydrophobic bacteria that could easily attach to these surfaces, or it will just happen. For example, in some already existing experiments on the field, people have realized that the first one to attach are diatoms, and then a succession of other, let's say, genus or species is coming up. So I'm not sure if biofilm formation, I still don't feel like that bacteria are selecting the surface like I want that surface. And it happens to us in our systems that are totally different than plastics, that the pre-growth conditions are also helping enhancing or inhibiting attachment to a certain surface. So my second question is, how do you feel of the carbon metabolism of these microorganisms? Because one thing is to attach to a surface and to start degrading it, and another one is to boost biomass, for example, with another external carbon source. Maybe these molecules are not really fitting into the, how to say, carbon metabolism for respiration, and then you still need to boost them with the external carbon. Is that a secondary metabolism?
So adding external carbon has not really been necessary once you have managed to enrich microorganisms which can do the degradation. In the beginning, we did add a little bit. Yeah. So we didn't add a lot. It was trace amounts. But if you don't add anything in the beginning, if you just have seawater, then this probably still happens, but it just takes much longer. Basically, you just need to get a little bit more biomass. Of course, then the question is, what are you going to add? Because everything you add is going to cause a massive perturbation in the community composition. So you need to maybe try out different additions, but again, if you add too much, then it's also not good because then they will prefer other carbon sources to this more difficult one. But yeah, after we have the established system, we didn't really need to add anything else. No, they are very happy just consuming the plastic as the carbon source. As I said, not all of them can consume everything. But yeah, we didn't really have any recalcitrant oligomers, for example, that could not be degraded or such. Yeah.
Did you try the changes on the microbial composition according to different, if you have a consortium already established, if you grow them on different types of, or sort of the same material, but with different rugosities or different sizes or just different composition, do you see that some microbes are enhanced or inhibited in the population?
What we try to do is we try to feed the monomers of the plastic, and this is massively different. So the microorganisms that then dominate the consortia are those that you can even barely detect when you give the plastic itself. So it does cause a very big change in the community composition itself. Adding plastic in different forms, such as flakes, powder, and such, does not make a difference. But the films are degraded much faster. You would think that actually a bigger surface area would be faster degradation, but I think this is actually maybe more favorable for biofilm formation because, for example, powder took much longer to degrade and to be converted into CO2 than the plastic film itself. I cannot account for all possible causes for this. There are so many things that go into processing the film itself. For example, if you're blowing the film, they put some substance on the surface to prevent it from sticking. Even that can make a difference if it's going to be degraded well or not. But this is the experience that we made.
And have you tried removing the planktonic population during the degradation?
It doesn't really make much difference because, as I showed before, there are no microorganisms which only exist in the biofilm or only in the plankton. So if you remove the film, just wash it and then put it in new medium, you still have the same community. It just equilibrates after a while.
Okay, thank you.
Any more questions? Okay.
About these two isoenzymes you found, we observed the same thing with the thermobifida, and one is both expressed in the presence of the plastic, and one is less active than the other, so similar. And then we looked at the surface of these two, and there are differences. We observed that we see some hydrophobic patches on different sides of the protein away from the active site. So we think it has to do with binding of the substrate, which is different. And the explanation may be that these two enzymes are just for another purpose. So they have some activity on the plastic, but in nature they do something else. So that's why they're there. Because it's very similar to what you described.
Yeah, okay. That's what I was thinking.
Thank you for a nice talk. So I'm trying to reconcile how you started the talk, which was a lot of misconceptions about biodegradation, and you showed this nice schematic of the ocean and the marine environment and all the different ways that the environment can be variable. And then if I understood correctly, you literally scooped some seawater from one location. So did you get lucky? Is that because you were at the interface with land? What if you took a community of organisms from the deep sea or from the oligotrophic dryer? I mean, isn't the community of microorganisms going to strongly influence the outcome of this experiment?
So first of all, the study turned out to be a lot of fun. So we did take a lot more samples from different geographic locations, and we tried them on different kinds of plastics. And the key is always to add sediment. If you just add water or open water, so just samples from the open water column, I think you just don't have enough microorganisms which prefer growing attached to surfaces. And I never succeeded in growing this kind of consortium just by using open seawater. It might also have to do with the biomass, just because the microbial biomass that you start with is so much less in the open ocean, like water situation. But I think, as I said, it also has to do with the lack of attachment specialists. I didn't try to take deep sea bacteria. I was asked if I want such samples, but to grow these you need pressure chambers, and I just don't have them. So I didn't really do this. Where you take the samples is definitely going to make a big difference. And the one thing that we should be clear about, the study that we did, although it's a nice demonstration, it does not accurately represent what is going to happen once the plastic is in the sea. In the sea, for example, okay, plastic is carbon, like there's nothing else. So to degrade carbon, you need nitrogen and phosphorus. In the sea, depending on where you are, these might be limited, for example. Maybe the temperature doesn't play, it does play a big role, but maybe it takes longer, for example, if you're in a colder place, although ambient temperatures also work. So limiting factors and such will play a role in how the plastic behaves once it's out there. This is why we also need to do field experiments, and any standard method that is developed has to, although the industry doesn't like it, it also has to include field experiments or mesocosm experiments, so something other than just a shaking flask. So what we do is great. I love my bacteria, but I cannot promise that if it falls into the sea, it will also degrade in 6 days. I can probably say it's not going to, but even if it degrades in 15 years, it's still an improvement, no? I don't know. Like, or maybe it is worse. I don't know. Maybe the degradation products are bad for some organisms. Maybe some bird is going to ingest this, and rather than just getting out, it's going to be digested into some oligomers, which are harmful for this bird. I don't know.
Yeah, I agree. Those are appropriate words
Like-
of caution. Yes.
So the thing is, yeah, exactly. So we don't know. We need to do more research, and this is why I wanted to just leave the controversy behind and maybe get some more information. And also, of course, plastic is carbon, and plastic is also a very good carbon sink. The plastic is in the ocean and it's inert, basically. If it is degraded, it turns into CO2, and CO2 is not really popular at the moment either. So these are not good options, but it is just another way to curb the problem, basically.
Can I ask one small follow-up? Does PBAT sink in water? Is it more dense than water?
It does sink after a while, yes.
All right, if there are no more questions, we are done with the symposium. Thank you, Vasak, for the presentation. Thank you to all of the attendees for holding up until this late. Well, it's not that late. We're just 15 minutes late. But yeah, thank you so much for everybody for coming, either in the morning session or in the afternoon. We have a coffee waiting outside, so you're more than welcome to enjoy it. And again, yeah, just to acknowledge, we did this with funding from the Office of Research from the Catholic University of Chile, Pontificia Universidad Católica de Chile. We also did it with support from the Millennium Institute for Integrative Biology, the Institute for Biological and Medical Engineering, and from a joint project with Brazil and a NEET FAPES project between the research agencies from both our country and Brazil. Thank you so much for coming. We're going to take a quick picture here. So if you just join me on stage, we will take a quick picture, and then we hop off the building for the coffee break. Thank you so much.